I'm Jim Baxter, and this is Lecture 8, Describing Populations for General Ecology. So, today we begin our discussion of ecological populations. Now, we've of course talked about populations a little bit during our discussion of evolution by natural selection, haven't we? The notion that the unit of evolution is the population. That is, it's a population that evolves over time. Of course, there's a reason for this, and that is that a population is a distinct unit in ecology, with individuals that occupy the same location and interact with one another in a variety of ways. This ecological unit, of course, is also dynamic, and when I say dynamic, I mean it's constantly changing over time. From the standpoint of evolution, which we've been discussing, that change occurs as changes in allele frequencies over time. But the population is a dynamic unit in more ways than that. It's ever-changing in the number of individuals within the population, its demographics, its range and distribution, immigration, emigration, and so on. And populations vary dramatically in these ways, depending on which species we're talking about. Today's lecture focuses on how ecologists think about populations and how they describe their various properties. But before we start, we need to define the population. So, what is a population? Well, a population is a group of interacting individuals of the same species that inhabit the same area. But species to species, this, the amount of, the area that they inhabit can be very, very different. For example, the apple scab fungus shown here, is a fairly small species, a microbial species, and it's a microbial pathogen on apples. So, the range over which this species occurs is very, very limited. Compare that, of course, to, a mammal like the Florida panther and its range. This map shows sort of southeastern United States. The green area is the historic range, and the yellow area within that is its current range. Obviously, this species occupies a much larger area than the apple scab fungus. In California, another example that you're familiar with from our field trip is gray pine. And this is a map of California showing in orange the geographic range of gray pine. Notice that it's limited to California, so it is one of our California endemics, and that it also occupies a very large range, much larger than many other species, such as the scab fungus that we just saw. And over even larger ranges, such as the gray whale shown here, and its migration patterns, it can occupy areas across broad latitudes of the Earth's surface. So species that are interacting with one another inhabit very, very different spatial scales. And where you find a population is, of course, affected by environmental factors, both abiotic factors, that is, the non-biological or physical factors, and biotic factors that can strongly influence where you find individuals and how many individuals in a population you do find. Some of the abiotic factors we've talked about. These are temperature and precipitation, but also, of course, light, the kinds of soils that are present. Harken back to our field trip when we talked about serpentine soils and how striking the difference was between the different types of vegetation on those two different soil types, on versus off of serpentine. Wind, etc., and other abiotic factors can really have a very strong influence on where you find individuals of a species. Biotic factors also can be very, very important as well, and we've talked about some of these, competition and predation, a little bit if you think back on the example when we were talking about niches, and we referred back to the barnacles, the balanus and thalamus. Those two species were in direct competition with one another, and that competitive interaction strongly influenced where you found one versus the other species. So the distribution of the populations of those species on those intertidal bluffs, intertidal zones, were strongly determined by competition. Predation, of course, as you might imagine, can also have very dramatic effects, as well as dispersal ability, the kinds of pollinators that are available to plants, pathogens, etc. So where you find a population is dependent upon, a complex combination of all of these factors. Populations occur over what's called a geographic range. That is the area over which all individuals of the species are found. The geographic range is often illustrated on a map that shows an outline of where individuals of the species are found. Of course, not every single location within any particular geographic range is occupied by individuals of the species. This is because even within that area, conditions vary and may or may not be suitable for the species. Geographic range is often determined by abiotic factors, such as temperature and precipitation, like we saw before. A good example of that is in several species of Encelia, a genus in the sunflower family, and these species have different geographic ranges in Southern California, Mexico, Nevada, and Arizona. In the far west, or the southwest of the United States, and along the coast, Encelia californica is confined to a narrow band along the coast, as you can see in the red shaded area on the map on the left. Whereas Encelia actonis range is farther inland, where conditions are drier and somewhat warmer than Encelia californica. Farther inland, a different species, Encelia farinosa, and the other Encelia fructescens inhabit areas that are much hotter and drier than the others. Okay, so if geographic range tells us about the overall range of a population, how do we describe its pattern of occurrence within its geographic range? When we look within the geographic range, we see that individuals of a species don't occur everywhere within that range, as I mentioned earlier. At this spatial scale, we refer to this as the distribution of a population. A nice example is that of Clematis vermontii, a plant species restricted to limestone outcrops in the Ozarks of Arkansas and Missouri. Now, an outcrop just simply refers to the underlying parent rock sort of exposed onto the surface. So, kind of like what we saw on our field trip with serpentinite, limestone rock gives rise to a certain type of soil on which certain species have become adapted, like this clematis. In the various maps in the slide, the population of clematis is shown at different spatial scales. So let's start with a map showing individual plants in the population and work up in scale from there. So the one with the individuals is shown in the lower left in panel E. Now each dot represents a single individual within a population, or actually within a subpopulation. A subpopulation within the larger population shown in panel A. Notice, and this is shown in D, that the subpopulations in this map all occur within a defined area. This area corresponds to the location of the limestone outcrop to which this particular species is adapted. Clusters of subpopulations are occurring within this outcrop. If you were to draw a line around all these little subpopulations, this creates the map shown in C. So you can see the arrows sort of expanding the scales, looking closer and closer in. So now when we've moved from panel D to C, we see that outcrop with all the subpopulations in it is now shown as a completely shaded area. Okay. Moving up to a larger scale, the scale of the watershed, we see that there are multiple clusters of subpopulations that occur and are coincident with some stream courses that are running through the area in the watershed, which are along, the outcrops are kind of along that stream course. In the top left is the entire range of the species, but showing the distribution of population clusters within that geographic range. So this isn't this isn't like the map of the geographic range for Encelia before in the previous slide. This is actually showing those multiple clusters, for the entire population. So the important point of this illustration is that populations can be viewed at multiple spatial scales, from individuals to subpopulations to the entire range of the species. Okay, so at the scale of the individual, though, we refer to the patterning of individuals at this very local scale as dispersion. Okay, and this is determined by a number of different environmental factors, and we'll talk about this in a second. Dispersion is how species, individuals of species, are patterned in space, whether randomly spaced or clustered in some way. So dispersion is a topic we're going to spend some time on in lab this week. So I'm not going to really spend a whole lot of time right now. You'll get plenty of chance to work with these ideas in the lab. But on the left and center and right, you'll see three different dispersion patterns. And these are different ways that individuals can be dispersed in a population on the landscape. Now, there are various environmental factors that will give rise to these. In the situation where there's an absence of positive or negative interactions between species, that's when you're likely to have a random dispersion pattern. The panel in the center. Species are neither repelled by one another or attracted to one another. On the other hand, if you look in the left panel, we call that a clumped dispersion pattern, and that's where the individuals are clustering together, perhaps because there's a coincidence of resources that they're taking advantage of, and so as you look across the landscape, you see that the species or the individuals are clustered together, or maybe they're social animals and they have a certain social structure, and this causes clustering. Maybe they're grouped because of protection or because of the way the seeds are dispersed or something like that. So the clump dispersion pattern is a dispersion pattern where we have positive interactions between individuals in a population. On the other extreme, to the far right, is the regular dispersion pattern. And this is a pattern that is caused by antagonistic interactions between individuals of a species. And primarily this would be due to competition. So let's take a look at one particular example that is quite interesting. Creosote bush is a plant species that occurs in desert ecosystems, and its geographic range extends throughout Mojave, the Sonoran, and Chihuahuan deserts. And because it occurs in a desert environment, as you might imagine, it competes intensely for limiting sources of water. This has resulted in a very distinctive dispersion pattern for this species. A photo of individuals of creosote bush is shown in this slide, and look how the individuals of the species are spaced in this habitat. What kind of dispersion pattern do you think this population exhibits? Well, it's probably not too difficult to determine that, given intense competition for water, that individual plants might be in competition with one another and therefore have antagonistic interactions and perhaps exhibit a more regular dispersion pattern. So let's take a look at creosote bush a little bit further, because dispersion patterns aren't necessarily constant in time for a particular species. And creosote bush is one of those examples. So dispersion patterns can change, and let's just take a look. So in this, in the circle shown there, we have creosote bush shown as little green, bushes there, and it's showing that when they're young, when they're very small shrubs, they tend to show a clumped dispersion pattern. And this is because the way the seeds are dispersed for creosote bush, the seeds actually don't fall very far from the plant. So when it sets seeds and those seeds are dispersed, they fall nearby the parent. Once they germinate and grow, you get these little clusters of young, small shrubs that show a clumped dispersion pattern. However, over time, of course, because of intense competition for water, what we see is that as the plants grow, there's mortality of some shrubs and not others. So some survive and others don't. As the shrubs grow, the clustering becomes less, and so the dispersion pattern shifts from a clump dispersion pattern to a more random dispersion pattern. Later on, as the shrubs grow and competition intensifies, as they get larger, of course, they need more water, and their root systems extend further to get that water. And so competition begins to play out between individuals of the species, and so competition really drives a shift towards a more regular dispersion pattern. So this is a good example illustrating that creosote bush actually can have all three dispersion patterns showing up in its population, depending upon the time point, of when after it has established in an area and it's gone through these phases of competition, bringing it from a clumped dispersion pattern to a regular. Okay, so you might ask yourself, how do we know that it's competition that is driving this pattern in creosote bush? It's assumed that and these antagonistic interactions are due to competition for limiting sources of water, but how do we know that that's really the case? Well, Brisson and Reynolds in 1994 actually conducted a really ingenious but also terribly laborious experiment where they actually excavated and mapped 32 creosote bush root systems. That's shown in the left panel here. And so what they did was, based on that, they assumed, okay, so they asked themselves, what if there was no competition? How would we expect those root systems to grow? They assumed that just like you might start a bacterial colony in a petri dish, an agar plate with bacteria growing in it, if you were to inoculate that plate, how would the bacterial colony grow without interactions from any other colonies or any other species? Wouldn't you expect it to grow in a outwardly in a circular pattern? That's exactly what they predicted for creosote bush. If any individual creosote bush is not in direct competition with any other, what they should see is that each root system of those 32 creosote bushes should grow in a circular pattern. And so what they did was they mapped out for each and every one a circle around the base of the individual that extended the distance of the root system out. And then what they did was they mapped out how many areas were in direct overlap with one another. And what they found was that 20% of the area included extensive overlap, and that overlap is shown in the center panel in blue with the hypothetical circular root systems, and then in blue is shown the area that would be overlap of those root systems without competition. Then what they did was they mapped the actual root systems and calculated how much overlap there was in the actual root systems that they had mapped. And what they found supported this notion of competition, because only 4% of the area of the root systems actually overlapped. So what obviously happened was that the root systems were growing not in a circular pattern, but in complex non-circular patterns that were avoiding overlap with other root systems, avoiding direct competition for that limiting resource, water. So they were able to support their hypothesis through, again, a really ingenious but very, very laborious method. Okay, so let's look at another parameter of populations that ecologists care about. We just talked about geographic range, distribution, and dispersion pattern. Now, what about abundance? Abundance requires understanding something called population density. And what's shown here in this slide is a bunch of pink dots that represent individuals in a population. Now, what if we want to determine the size of that population, the total number of individuals in the population that would give us an indication of abundance? Well, one of the ways that we can do this, and this again, we'll be looking at this in lab. We'll be actually going out to measure the population size, density, and dispersion pattern of a species out along the American River. And so one of the ways that ecologists do this for stationary populations is that we can actually take multiple samples across the landscape and then determine what's called a population density. Once we know how many individuals there are per unit area, we can scale that up to the entire area within which the population exists and get an estimate of the size of the population. So what's shown here are 10 plots or quadrats that if we were to count the number of individuals in each one and take the average, that would give us an average density or a mean density. In this case, it would be 1.7 individuals per unit area. If each of these plots were, say, 1 square meter in area, to get the total population size, we would simply multiply by the total area. Now, of course, this is a rather crude method because we know that actually species don't occur evenly across the landscape, particularly in larger areas. So what we need to look at is the actual ecological density, all right, as opposed to a crude density. So what's the difference? Well, this image here shows an area that was a forested and mountain area that was logged, and there are some forest fragments remaining. What if we wanted to calculate the density of bird species? Let's say it's a forest bird, a bird that occurs in forested systems. Well, would we want to calculate? Would we want to sample plots throughout the entire area, including the logged areas? No. We would want to focus our sampling within the forested areas where we know the bird species occurs. This is called an ecological density because it's the population density within the habitat areas that the species actually occupies. Although abundance tells us about the number of individuals in a population, it doesn't tell us anything about the characteristics of individuals in the population. Why does this matter? Well, it matters because the relative proportion of individuals of different types, such as ages, can have an important impact on the growth of the population. Why is this? Well, the answer is that not all individuals contribute equally to a population. One of the ways individuals contribute differently to the population is in their reproductive potential, and therefore their contribution to population growth. Because populations have individuals of different ages, and different ages have very different reproductive potentials, age structure is a very important population parameter. Age structure can be represented graphically with an age pyramid, sometimes also called a population pyramid. Although a population's age structure isn't always pyramidal in shape, most populations tend to have more younger age classes than older ones. These diagrams provide scientists with a profile of the population's age structure. This demographic information about a population allows us to determine the reproductive potential of that population, and in turn to estimate current and future growth. Of course, this is a really important population parameter for ecologists as well as sociologists, for economic reasons as well. For our purposes, we're going to be thinking more about natural populations than human populations. But let's take, as an example, let's take a look at a human population. that we can identify with to give us a sense of how this works. So this can be seen in the slide here of the age pyramids, which show the age structure of Japan at two different time points in the past and then projected for 2050. Usually, a rapidly expanding population will have a larger reproductive, larger number of reproductive individuals, whereas stable populations will show a more even distribution of age classes. Rapidly declining populations will have large older cohorts, or large numbers of older individuals. So looking at this slide, in the left panel in 1950, you can see that the age structure or distribution of individuals in the Japanese population showed a much more classical age pyramid structure with large numbers of individuals in the younger age classes. And if you take a look at this, essentially what you're looking at is on the bottom, on the x-axis, millions of people, and then on the y-axis going up, the age groups. And each bar on each side represents the numbers of males, usually indicated here on the left, and females on the right. And the percentages show the different percentages of the populations in the different age classes. So in 1950, clearly the population was fairly rapidly growing because it had a lot of younger individuals, many of whom were in reproductive age, contributing to population growth. In 2007, however, this changed, and all of those individuals in those younger age classes grew up, of course, and contributed to the 15 to 64-year-old group, shown in blue there. But those individuals did not have as many offspring, and therefore there was a less contribution to younger age classes, and so the population growth started to slow. The projection is, if current trends continue of low reproductive rates in the population, that is projected to result in a more, an upside down pyramid structure, where the population is dominated by older age classes of non-reproductive age, with very few individuals in younger age classes of reproductive age. So, because reproduction is restricted to some age classes, and mortality is more important in others, the relative proportions of each age group will strongly affect, how quickly or slowly populations grow. There are three ecologically important age classes, or stages. The first one being pre-reproductive. So these are the age classes where individuals are pre-reproductive. They are not currently reproductive, and so those individuals are not contributing, not yet anyway, to the growth of the population. Reproductive cohorts or groups, age classes, are those that are contributing strongly to the growth of the population, and post-reproductive are those individuals who are no longer reproductive, obviously, and not contributing to population growth. So let's take a look at this phenomenon in an ecological population. Okay, these are population age structures for two species of plants, white oak and cottonwood, and it's illustrating the effect that age structure can have on the growth of populations. So in the white oak population, you can see that the age structure shows that older trees are being replaced by younger, because you have many fairly young trees, that is 0 to 50 years old. So these are fairly young reproductive individuals that are supplying new individuals to the population. So this population is dominated by young individuals and has a very strong potential for growth. Okay, so older trees are being replaced by younger. In the cottonwood example, however, there are very few young. Older trees are not being replaced by younger trees, And so the absence of these young trees suggests that the population is probably declining rapidly, and that older individuals will not be replaced. What it looks like, could have happened here in this population is that there was some maybe a mortality, event, or something that restricted reproduction about 40 to 50 years ago, and so that's playing out. But the point here is that without younger reproductive individuals, the population will not be able to persist, and likely for cottonwood, this population is declining. Another population parameter of interest to ecologists, and that relates to populations, is what's called dispersal. That is population dispersal. This is different from seed dispersal and that sort of thing. This is a population phenomenon in which individuals of a population colonize new locations and establish new populations, and therefore expand their range. A really fascinating example is the example of the European starling, which was introduced into the United States in the late 19th century, and rapidly spread across the United States. It turns out at that time of the introduction, a group called the American Acclimatization Society was working on a project to introduce to the U.S. every bird species mentioned in Shakespeare's scripts. Okay, so you can imagine. Shakespeare was quite a birder. He mentioned some 600 different bird species in his scripts, and so this society felt that its devotion to Shakespeare required it to bring over a whole bunch of exotic bird species. The European starling was among them, and a Bronx resident, actually it turns out, named Eugene Schiefelin, it seems, was particularly responsible for many of the starlings' introductions. So the society released some 100 starlings in New York City's Central Park in 1890 and 1891, and by 1950, the starlings could be found pretty much, almost coast to coast, up into the Canadian Hudson Bay and south into Mexico. The maps here kind of give you a sense of that colonization pattern. The first panel in the upper left in 1918 shows the geographic range of the bird in red in the Pacific, in the northeast there, and then the dots, population centers where the bird had dispersed, began to form new colonies, but hadn't completely covered that geographic range quite yet. If you look forward to 1926 and then 1949, you can see that those initial colonizations resulted in the expansion of its geographic range into these new locations, and eventually after 1949 or 50, it slowly but surely colonized the entire United States. And today the map of the U.S. is virtually all red because the European starling has become naturalized here, and now its geographic range covers the entire United States. So dispersal as a phenomenon is a tremendously important one for species, and it does allow it to expand its range, to occupy new habitats, where species like the European starling and others are introduced into new habitats because of the lack of pathogens and predators and that sort of thing. They can actually establish quite rapidly into new habitats. So population dispersal is a really important phenomenon in populations in terms of colonizing new habitats and expanding their range. Finally, when we think about populations and individuals moving around in populations, we know that populations occur in what we call subpopulations, and individuals move among these subpopulations. When considered together, we refer to this as a meta-population. We'll be talking about meta-populations a little bit later, but I want to just emphasize that between these subpopulations, populations are connected. They're connected by immigration, that is, individuals moving into the population, into a subpopulation from another subpopulation, and emigration out of a subpopulation into a different subpopulation. An example that's shown here is snail kites. This is a bird species that occurs in southern Florida, and the map here is just showing the different subpopulations as the major nodes, and then the movements among those subpopulations that have been documented by this bird. So these individuals are moving among subpopulations. It occupies its natural habitat or wetland areas, and so these subpopulations occur within these wetland areas, and but there's continuous movement among these subpopulations. And of course, as a consequence of that, individuals are resupplying subpopulations with new individuals, and also thinking back to evolution and gene flow. Of course, those movement patterns are also exchanging alleles among individuals across these populations. So why do individuals move? Well, we'll be talking about that a little bit later on down the road, but certainly some of the factors that can influence whether or not an individual moves could be, the population density has increased so much that competitive effects are playing out, and it ventures out of the population to find new habitat and new resources, maybe new nesting sites or new mates. The effects of these movements, as I mentioned, are resupplying subpopulations that might be otherwise small or go locally extinct by neighboring populations. So those movements can actually keep those small subpopulations intact, so they don't go locally extinct. And of course, gene flow and the mixing of alleles across the individual subpopulations. We'll be talking about a number of these things going forward. So, but this wraps up the lecture for today, lecture 8, and we'll continue on next time with population growth.